everyone, welcome back to my channel. If you are a returning viewer, it's great to have you here again. And if you're new, welcome. I create bioinformatics videos and step-by-step tutorials, hoping to make and break down complex topics simple into simpler and accessible chunks for everyone to understand. So thank you for joining me today for another exciting video today. So in today's video, I want to discuss and talk about a situation some of you might have already encountered. So let's say you were asked to process one sample and you were given, you were pointed to a location where the files from the sequencer were uploaded. And you think in your mind that it's going to be fairly straightforward to analyze one sample because it would be a one FASTQ file if it's a single-ended read, or two FASTQ files for paired-end reads. But then you go to the folder, you look at the files, and you see these bunch of files here, something similar to these files, and there are a bunch of questions as to why are there so many files for one sample, how to make sense of these files, what are these numbers underscore these letters, and why are there so many files, and more importantly, how to handle these files. Should I merge them? Should I not merge them? What do they mean? Before we jump right into understanding what these files are, how to make sense of them, and how to handle them, it is important for us to understand some core and basic concepts in sequencing, in this next generation sequencing, for us to make better decisions about when to merge them, how to handle these files, and how to process them. So we first start by understanding what is library preparation. So library preparation is the process of turning your nucleic acid, that is your DNA or RNA, into something that the sequencer can read. And it usually involves steps like nucleic acid extraction, fragmentation, end repair and A-tailing, adapter ligation, enrichment, and quality control and quantification. Followed by that, there is another terminology which you would have heard a lot often when dealing with sequencing data, which is barcodes. So barcodes indexes are usually short DNA sequences that uniquely label each sample. So if you sequence multiple samples in one run, and this process is called as multiplexing, barcodes are unique and they help you tell which read is coming from what samples, and will help you segregate your reads based on the sample that it's coming from. An important question here could be, can the same sample undergo different library preparation? The answer is yes. And the next obvious question would be, why would you want different library preparation for the same sample? The reason is there can be various sources of technical variation, and hence you would want to create technical replicates. So basically, you would have the same sample, you would process it using the same steps, but you would just have different libraries prepared. So this would help you assess the technical variation in different steps that I mentioned, like fragmentation, adapter ligation, PCR amplification, or even during capture efficiency. So if one preparation fails or is insufficient, the labs can also repeat the library preparation or pool two library preparations to boost the yield. Another reason of creating or having a different library preparation for the same sample would be to test the batch effects, like kit comparison and automation consistency to assess that. And lastly, another reason you could have a different library preparation could be a different sequencing goal. So to have different coverage depths or have long read versus short read data, etc. The next concept or terminology we must be familiar with is a flow cell. So a flow cell is where the sequencing happens. It's a glass slide that's divided into lanes. These are physical partitions that process DNA independently. And these lanes can run the same or different samples. And this chip essentially, or a glass slide, is eventually put into the sequencing machine, which sequences the DNA and outputs the reads. So one of the question would be, why run multiple samples on the same flow cell? There are a couple of reasons you would want to do that. One could be maximizing flow cell capacity. So modern sequencers have high throughput. If a single sample doesn't need the entire capacity, multiple samples can run together to maximize its use and reduce the cost per sample. In addition to that, pooling multiple samples on the same flow cell allows shared sequencing run, which is more economical than running one sample per flow cell. The next reason could be multiplexing. By adding unique barcodes to each sample, many samples can be sequenced in parallel and later separated bioinformatically. So this reduces the turnaround time and increases the throughput. And lastly, it also allows for more even distribution of DNA across the flow cell, potentially reducing variability due to lane differences. On the flip side, one could ask why run the same sample across multiple lanes? There could be a bunch of reasons for this, but a few reasons include reducing lane bias. So, running the same sample across multiple lanes can help mitigate or remove the technical variation between lanes, improving data reliability. In addition to that, combining data from multiple lanes can increase uniformity in coverage and reduce dropout or under sequencing in any single lane. It can also allow us to distributing a sample across multiple lanes can allow us to capture more reads, which is especially important for applications requiring higher depth. And lastly, if there is a problem that occurs in one lane, the data from other lanes still provides useful information. So going back to my point on running the same sample across multiple lanes allows us to collect more reads and hence achieve higher depth. This it's important for us to clarify this point, especially when a library preparation document promises a 30x coverage per library. It is important to question whether that means one library per lane yielding 30x from a single lane or pooled across multiple lanes, that is 30x in total, which provides us a 10x coverage on an average per lane. So it's always important to confirm whether the coverage that is expected out of the library is per lane or is across the total run. Although the yield remains the same, but having no redundancy is a big disadvantage, because no redundancy would lead to data loss, and multi-lane splitting is more preferential because it provides fail-safe redundancy, allows you to perform lane-specific quality control, and reduces the lane bias. And there is risk when putting all the data into one lane. It could result in the data loss due to lane failures. It could result in uneven base quality, over clustering, or barcode crosstalk. Now that we have a fair share of understanding of the basic next generation sequencing concepts, the terminologies, coverages, and the concept of splitting data across multiple lanes in a flow cell, let us take a look at a real life example. In this example, we will be looking at different technical replicates that have unique barcodes that have been pooled together, multiplexed, and sequenced across multiple flow cells, and eventually demultiplexed into separate FASTQ files, which have been split across multiple lanes into chunks. So basically, I want to show you how the data is organized, how to make sense of that data, and also provide suggestions on where to merge them and when to not merge them. So the example that we will be looking today is from Genome in a Bottle. So Genome in a Bottle is a project that is led by the US National Institute of Standards and Technology that develops highly accurate human genome reference materials and benchmark datasets. So essentially, it's a gold standard set of genomic sequences from specific human samples that you can use to benchmark or validate your workflows, your tools, and also assess variant calling accuracy. So let us navigate to Genome in a Bottle dataset that I wanted to show you. So I'm going to go into the Genome in a Bottle GitHub instead and get the data indexes. So essentially, this page lists down all the sequencing that have been performed. They have been sequenced on various sequencing platforms. And essentially, there are different set of data sets. This one that I'm looking at is the Ashkenazim trio, and I'm going to be taking a look at the data from the sun. These are the data set. These are publicly available that you can use for your analysis. So I'm going to go over the link provided here. And as I said, there is a folder, one for each of the different types of sequencing that have been performed. So I'm going to go into one of them. I'm going to go into the one that has HiSeq 300 FASTQs. So when you go inside this folder, but before I go inside and start explaining the various folders and the organization, there is a README here, and you will find this README in every folder. So essentially, there are a bunch of folders here, a bunch of runs, and also you have FASTQs, you have BAMs, and you have a .text file. This is an important file because this README describes the library preparation and sequencing that have been performed for the sample or the data that is present in this folder. So it's very, very important for you as a best practice to always go through the library preparation and sequencing documentation. If this is something that is not provided by the sequencing core or anyone who is providing you for with the sequencing data, it's important to ask for this information, because a lot of the things or a lot of decisions that you make going forward will depend on this. So it's very important for the sequencing core or the group that has performed the sequencing or the person that has performed the sequencing to provide you with the complete information on the library preparation and the sequencing. Uh, since this is the large enough document, I'm not going to go into the details of each of the complete document line by line. That is something that you can explore on your own, but there are a few important things that I have picked out from this library preparation and sequencing and that I want to highlight that can help us understand the data a bit better. The documentation was pretty comprehensive. I just picked out the important points that we need to focus on to understand this data better. So the data source is from the National Institute of Standards and Technology, a reference material, and the data that we will be looking at only includes the HG002 data, that is the Ashkenazim Trio's sun, the data from the sun. The libraries, they mentioned that the libraries are prepared using 6 vials of the reference DNA. And there are 2 libraries that are prepared from each sample, which results in the total of 12 libraries. The size selection has been done with the magnetic bead purification. Adapter ligation, the indexes are added using the Illumina kits. Quantitative PCR has been used for concentration measurement. We should expect 20 to 30x coverage from a single flow cell, and the runs were designed to get the total coverage of around 300x. This is a pretty high coverage, and we should expect, if we combine all the data together, we should expect to get this high a coverage. In terms of the library preparation, Illumina TrueSeq DNA PCR free sample preparation kit was used, and the libraries were pooled and were run on the Illumina HiSeq 2500 machine with and that resulted in a paired adrenes of 148 base pairs. So going back a level into this folder, which has the FASTQs, here we see a bunch of folders. So essentially these are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. There are essentially 12 folders, and each of these folder corresponds to the data coming out of one flow cell. So there were 12 libraries that were pooled and were run across 12 flow cells. So each of the folder corresponds to the data coming out of one flow cell. And the way you read this is, the first part of it is the run date. So this one's from 2014, 05 is May, and the date is 28. So this corresponds to the run date. The next part of it is the sequencer ID. The next part of it is the run ID, and followed by that we have the flow cell ID. So looking at the flow cell ID, each of the flow cell ID is unique, which tells us that each of the data corresponds to a separate flow cell, and it contains, and when you click on this folder, there is another subfolder with the project in it, and then you will see all 12 samples. So essentially the 6 vials, so 6 technical replicates, which have two unique library preparation for each of these. So if you go inside one of these folders, you will get to see the FASTQ files. So the first part of it corresponds to the sample or the replicate identifier. The second part of it is the unique index or the barcode that was used to specifically identify the reads that are coming from this particular sample or replicate. The next part of it is the lane, so on which flow cell lane it was run. The R1 and the R2 indicates the forward or the reverse reads in a paired end reads, and 001, 002 indicates that the forward reads were further chunked into individual FASTQs. That is, this is the first chunk and this is the second chunk with with this sample, with this barcode ID on the lane 1 with forward reads that have been chunked into 2 FASTQ files. So that's basically how you interpret each of these FASTQ files and make sense of them. So going back one level, if you go to the second sample, you should see a different barcode ID. So no two technical replicates or no two replicates should have the same IDs or the barcodes. They should have separate. That's how you would be able to pool them, run them together, multiplex them and demultiplex them, and identify which reads are coming out from which replicate. So similarly, if we go a couple of levels back and if we go to the second folder that corresponds to the second flow cell, again the files are organized in the same way inside the project, and again you'll be able to see the 12 folders corresponding to 6 vials and 2 library preparation per vial. So hence you see the same set of namings for the folders, and you should see the same index and barcodes corresponding to that particular library preparation. So if I were to show this to you visually, this is how it would look like. So basically we have our proband or the data from the sun, and that from that we have 6 vials being collected, and there are 2 libraries being prepared from each vial. So total we have 12 libraries. You will notice that there is a unique barcode for each of the library, and all these libraries are pulled together and they are run across 12 flow cells. So the idea behind doing this is to achieve high coverage. So if you remember from the library preparation document, they did mention that we should expect around 20 to 30x coverage from one flow cell. So if you multiply that by 12, on an average, you achieve a very high sequencing coverage of around 300x. So this was the approach used. So instead of creating new libraries for each flow cells, the same 12 libraries were pooled and sequenced across multiple flow cells. So after each sequencing run, demultiplexing separated the reads by the index or the barcodes, and hence you see the same 12 sample folders in every run across each flow cell. So to give you further intuition, the A1 sample A1 from each run contributes only a part of the total data for A1 library. So if you were to construct the full data for A1 library, you would have to combine the FASTQs corresponding to A1 from each of the flow cell outputs to get the full data for that library. Now that we understand why there are these different files that we see for each sample and how they are organized and how to understand them, the next question is how do we handle these multiple FASTQs? So it depends on the context and the kind of the data that you're dealing with and ultimately the goal of the analysis. But here is a generalized best practices to follow, which holds true in most cases. So if you have the same sample which is prepared using the same library preparation method and spread across multiple lanes, then it makes sense to merge the FASTQs before mapping. If you have the same sample but you have different library preparation, it's better to merge them after mapping or aligning with the genome, the reference genome, and merging the BAM files later while maintaining the read group tags. If you need to perform the lane level quality control, it's better to use the FASTQs from each lane and map them to the reference genome separately and then merge the BAM files and perform the quality control. If you are dealing with single cell RNA sequencing or multiplexed RNA sequencing, then it's better to demultiplex the first as a first step before you perform any further steps. And if you have different samples with different library preps, it is better to not merge them and treat them as separate replicates. Because if you have different samples, it could be either different biological replicates or completely different samples, and you would want to retain the biological variation in them and not merge them. So these are just very basic uh, guidelines to handle multiple FASTQs, but ultimately the decision to merge and to not merge would depend on the goal of the analysis and the data that you have. So the last part is how do we merge these files? So if we have to merge the FASTQs pre-alignment, then we would simply cat it or combine it according to the the reverse or the forward reads across all the lanes. So we could basically just have like a combined FASTQ for the forward reads and the reverse reads, which are combined across the multiple lanes. If you have to merge the files or the aligned reads after alignment, you can use SAMTools merge or Picard jar. Both these tools would retain the read groups, so eventually down the line you would be able to track which reads are coming from which samples or replicates. So that brings me to the end of this video. I will put in all the relevant links in the description section below. So you could navigate to the same links for the Genome in a Bottle dataset that I did today. And you could take a look at the data and these files yourself. So if you enjoyed this video, please consider giving it a thumbs up. Don't forget to subscribe to my channel for more bioinformatics related content. If you have a question or a suggestion or a topic you would like for me to cover, please leave that in the comment section below. Share this video with your co-workers and friends if you found it helpful. Please stay tuned for the new videos. I really appreciate your support and I'll see you in the next video.